Welcome to Lecture 5, The Role of Natural Product in Drug Discovery and Development, Part 2. In this session, we will explore the journey of how compounds become drugs, the processes involved from discovery to approval, and the intricate ways in which these drugs interact with biological targets. First, we have to know what kind of compounds become drugs. Drug discovery is a bit like panning for gold. You have to know what you are looking for before you start searching. Among all possible delivery methods, oral administration remains the most preferred route. Most successful oral drugs follow a principle known as Lipinski's rule of 5. For a compound to be considered orally active, it should have a molecular weight of no more than 500 daltons, fewer than 5 hydrogen bond donors, fewer than 10 hydrogen bond acceptors, and a log P value between -1 and +5. Meeting this criteria indicates that a compound will have good permeability and solubility, key factors for success as an oral drug. The process begins with identifying the biological target, most often a receptor or enzyme that is linked to a disease. Scientists then screen chemical or biological compounds against these targets using specific assays to find promising leads candidate. Today, modern tools such as genomics, proteomics, synthetic chemistry, recombinant DNA technology, laboratory automation, and bioinformatics have transformed the landscape. Drug discovery is no longer the sole domain of synthetic chemists. It now requires the expertise of molecular biologists, engineers, statisticians, and even computer scientists. From concept to market, developing a drug takes an average 10 to 12 years, a timeline stretched not only by the development of successful drugs but also by the cost of failures along the ways. In the preclinical stage, compounds are tested in vitro in the laboratory and in vivo in animals to evaluate their pharmacological effects, safety, and delivery potential. This phase follows strict good laboratory practice guidelines. If the compound passes, it moves to the clinical stage where human trials assess safety, efficacy, and doses. Clinical research adheres to good clinical practice and must meet the regulatory requirements of authorities such as the US FDA, the EMA, or Japan's PMDA. Manufacturing for trials follows current good manufacturing practice to ensure quality and consistency. Finally, a marketing application is submitted for approval, and even after launch, the drug's safety and quality are continually monitored. Drug discovery today follows multiple approaches. The irrational approach relies on empirical screening of compounds, often from natural sources. The rational approach designs drugs based on the three-dimensional structure of the target. Newer strategies include antisense technology and RNA interferences, which act at the genetic level, as well as the development of biologics, gene therapy, and stem cell therapy. Each represents a unique way of addressing disease at its root causes. What is the irrational approach? This approach is the historical method of discovering and developing drugs. It involves empirical observation of the pharmacological effects from screening of many chemical compounds, mainly those from natural products. The active component that gives rise to the observed effects is isolated. The chemical formula is determined and modifications are made to improve its properties. This approach has yielded many drugs that are available today. Next one is the rational approach. This approach requires three-dimensional knowledge of the target structure involved in the disease. Drugs are designed to interact with this target structure to create a beneficial response. Having been established during the past 3 decades, the rational approach has become an important field in drug discovery. Next one, that is the antisense approach. This is a relatively new approach and it requires the modification to oligonucleotides that can bind to RNA and DNA. Antisense drugs are used to stop transcriptional from DNA or translational from RNA pathways from proceeding and so interfere with the process of disease. Next, RNAi approach. This uses short interfering RNA, siRNA, sometimes called small interfering or silencing RNA, to interfere with the expression of a particular gene. The siRNAs are double-stranded RNAs of 20 to 25 nucleotides. It is envisaged that if the biological pathway of a disease is identified, siRNA could interfere to turn off the activity of the gene involved in the pathway and provide therapeutic effect. Next one, that is the biopharmaceutical or biologics. These are mainly protein-based drugs in the form of antibodies, vaccines, and cytokines. Their discovery generally starts from an understanding of the biological mechanistic pathways that cause specific diseases. Manufacture of these drugs is based mainly on rDNA technologies using living organisms such as bacteria, yeast, and mammalian and insect cells. Gene therapy. This therapy is based on remedying a disease gene by inserting a missing gene or modified gene in the cells. This is an emerging field that raises many unresolved ethical considerations. The cells with the diseased gene are taken out of a patient, modified outside the body, means ex vivo, and then reinserted back into the body. In the case of missing gene, a copy of the new gene is inserted into the patient's cells. The aim is for the inserted gene to influence the disease pathway or to initiate synthesis of missing proteins or enzymes. Last one, that is the stem cell therapy. The aim of stem cell therapy is to grow body parts to replace defective human organs and nerves. Stem cells are harvested from very early embryos or umbilical cord blood. Because of the very young age of these cells, they can be directed to grow into organ tissue to replace diseased tissue. Recent research has enabled adult stem cells to behave similarly to embryonic stem cells and differentiate into different cell types and tissues. The stem cell technology can provide an alternative to organ transplants with perhaps lower rates of rejection than the current practice of obtaining organs from a donor. Stem cell therapy using germ cells involves cloning, and there are strict regulatory guidelines on how research is to be conducted. For a drug to work, it must interact with a specific biological target, much like a key fitting into a lock. Historically, the irrational approach, trying many keys until one fit, was common. The rational approach, however, starts by studying the lock first, then designing the perfect key. Today, advances in genomics and proteomics allows us to identify and validate disease targets before designing drugs. Scientific innovation alone is not enough. Pharmaceutical companies also consider market potential, patent strength, regulatory environment, and their own core competencies before investing in drug development. This ensures that resources are directed towards projects with the best chance of success in both scientific and commercial terms. The important factors to consider are the following, that are the market potential, patient intellectual property portfolio, or competitive forces and regulatory status, core competencies, then the pharmaceutical organization needs to project the expected returns from its drug development investment and assess the competitive factors and barriers, including government regulations, before deciding which drug to develop. Next, the pharmaceutical companies have to be continuously vigilant and forecast the future directions of drug developments and regulatory requirements. They have to use their core technical competencies to deliver a pipeline of products to remain competitive and profitable in the long term. First of all, I'm discussing with the natural products and their utility as therapeutics, advantages and disadvantages. Now it's the time for the methods of target identification. Radioligand binding was once the standard method for finding targets, but now DNA microarrays and in silico methods dominate. Microarrays allows scientists to measure gene expression across thousands of genes at once, comparing healthy and disease states to identify key differences. These findings can then guide drug design. There are a number of techniques used for target identification. Number one, that is the radioligand binding, was a common technique. Number two, the DNA microarrays. And number three, the in silico method. First of all, the radioligand binding. What is that? The classic method to discover drug targets or receptors is to bind the potential receptors with radioligands so that targets can be picked out from a pool of other receptors. Bound receptors are then separated from the radioligands, sequenced, and their nucleotide sequence is decoded. Potential drug molecules are then studied with these receptors or their nucleotide sequence to determine their interactions in terms of biochemical and functional properties. Then what are the radioligands? Ligands. What is the meaning of ligands? Ligands are molecules that bind to a target. They may be endogenous, that is produced by the body, such as hormones or neurotransmitter, or exogenous, such as the drug molecules. Ligands, the exogenous or endogenous, with high specificity for particular targets, are labeled with radioisotopes. The tissue cells known to contain the target are mixed with a known quantity of the radioligands. The target bound with radioligands can be isolated and its amino acid sequence determined. Next one, that is the DNA microarray. DNA microarray, also known as the DNA or gene chips, is a technology that investigates how genes interact with one another and how they control biological mechanisms in the body. The gene expression profile is dynamic and responds to external stimuli rapidly. By measuring the expression profile, scientists can access the clues for the regulatory mechanisms, biochemical pathways, and cellular functions. In this way, the microarray enables scientists to discover the target genes that cause disease. Then what is the microarray? To use the microarray, known sequence of short DNA is printed in a regular grid-shaped pattern onto a solid support of a membrane or glass slide. The mRNAs are isolated from the healthy and diseased cell. The mRNAs are used to generate complementary DNAs, means the cDNAs. Fluorescent tags are attached to the cDNAs and the cDNAs are mixed and incubated with the microarray support. Through a process called hybridization probing, the genes from the samples pair up with their complementary counterparts on the solid supports. When the hybridization step is completed, a scanner, the laser beam, and camera is used to capture the fluorescence image of the array. The expression levels of gene from the diseased and healthy cells can be deciphered by comparing the intensities of the red and green fluorescence. For example, if the disease causes some types of genes to be expressed more of these genes will hybridize with the DNA on the solid support, providing a greater intensity of red fluorescence than of green. In this way, disease targets can be identified. Now come to the point of express sequence tags and in silico methods. Are short nucleotide sequence of cDNA with about 200 to 500 base pairs. They are pairs of the DNA that code for the expression of particular proteins. ESTs sequencing provides a rapid method to scan for all the protein coding genes and a tag for each gene on the genome. The scanning of nucleotide sequences is achieved through in silico, the computer method. The promise is that all the proteins, even those with sequences that appear considerably different, can be members of families sharing essentially similar structure, and scientists can carry out their searches on database. Each EST of interest can be compared with sequences in proteins and the degree of match determined. A technique called threading is used in this type, which involves using data on 3D protein structures coupled with knowledge of the physicochemical properties of amino acids to determine if the amino acid sequence is likely to fold in the same way as the sequence for which the structure is known. In this way, more information about the putative target protein can be assessed. After the target identification, we need to validate that target. Validation. For this reason, some questions that arise in the target validation have to address. That is what is the function of the target? Which disease pathway does the target regulate? How important is this disease pathway? What is the expected therapeutic index if a drug is to interact with the target? The validation can be done in vitro, in vivo, using animal models, or increasingly in silico through computational method. Collaborative efforts such as the Accelerating Medicines partnerships are helping to speed up these crucial steps. Drug interactions with target. It should be clarified that targets identified using microarrays are mainly the genes that regulate or contribute to diseases. These genes targets give clues to the proteins that are affected. In most situations, it is the proteins or receptors that drug molecules are developed to interact with to provide the therapy. There are exceptions are in case such as the antisense and RNA interference drugs and gene therapy where the nucleotide, RNA and genes are targeted. When presented to the target, the drug molecules can elicit reactions to switch on, switch off, or alter certain biochemical reactions. The main drug targets in the human body can be classified into three categories, that are the enzymes, intracellular receptors, cell surface receptors. The hydrophilic or water-soluble drugs do not cross membranes. They stay in the bloodstream for duration that are normally short, lasting in the order of seconds, and mediate responses of short duration. In contrast, hydrophobic drugs, being water insoluble, require carrier molecules for transport through the bloodstream. Hydrophobic drugs remain in the bloodstream and can persist for hours and days, providing much longer effects. When the action of a drug is to active or switch on a reaction, the drug is termed an agonist. On the contrary, if a drug switches off the reaction or inhibits or blocks the binding of other agonist components, onto the receptor, it is called the antagonist. When the interaction is with an enzyme, the term inducer and inhibitor are used to denote drugs that activate or deactivate the enzyme. It should be noted that the drug molecule, agonist or antagonist, receptor and cell membrane are in fact complicated three-dimensional structure. The analogy is that only certain keys can be inserted into the lock and activate or deactivate the lock. Some facts about interactions between drug molecules and targets to bear in mind: the binding is specific. Binding occurs at particular sites in the target molecule. Binding is reversible. Allostatic binding occurs between two molecules bind to defined sites on the target when the two molecules are identical. It is termed homotropic interaction. If the molecules differ from each other, it is termed heterotropic interactions. Binding is competitive when two defined ligand molecules compete for the same site. Then what are the types of interactions? Binding between drug molecule and receptor or enzyme is critically dependent on the shapes and sizes of the molecule. To deliver the therapeutic action, drug molecules with the right shapes and sizes have to be designed to fit into the binding sites or pockets of the receptor or enzyme. Another important factor is the nature of the coupling. Before drug can fit into the binding site, it has to overcome thermal and vibrational motions at the cellular level. The attractive forces must be strong enough for the drug to dock with the binding site. When molecules couple together, the type of bonding can be divided into covalent bonding and electrostatic interactions due to hydrogen bonding or van der Waals forces. The stronger the coupling between the drug and binding site, the more sustained is the interaction. Covalent bonds are strong bonds. Actual bonds are formed between the interacting molecules via the sharing of electrons. So there are two types of electrostatic interactions, that are the hydrogen bonding and another one is van der Waals forces. Hydrogen bondings are due to the attractive forces between the distorted electron cloud of a hydrogen atom and other more electronegative atoms such as oxygen and nitrogen. The attractive forces are weaker than the covalent bonds, but many hydrogen bonds can be formed in macromolecular protein molecules. Van der Waals forces are weaker attractive forces due to the attraction between neutral atoms. A third type of the interactions follows the hydrophobic effects. These are the result of non-electrostatic domains interacting. This type of interaction occurs mainly with the highly lipid soluble drugs in the lipid part within the cytoplasm of the cell. Biochemical reactions can release energy, exothermic, or require it, endothermic. Enzymes lower the activation energy, making reactions proceed more quickly and efficiently. In general, the two types of biochemical reactions, that is the exothermic and endothermic. Exothermic reactions are those where the energy states, the free energy labeled as G of the reactants, are higher than those of the products. They are energetically favorable. The next one, the endothermic reactions, are those for which the products have higher energy states than the reactants. They are energetically unfavorable. Regardless of whether the reaction is favorable or not, the reactants have to come together to close proximity to react. They have to overcome a potential energy barrier that may involve displacing solventing molecules around the reactants and reorientating the reactants. Enzymes bind to the reactants and provide an alternative mechanism of lower activation energy for the reaction to proceed. Hence, the enzymes speed up biochemical reactions that are otherwise too sluggish to advance. Now the time for the receptors and signal transduction. Cells communicate to coordinate the biological functions within the human body. If the biochemical pathway communication system is interrupted or messages are not conveyed fully, our body functions can go haywire. There are hundreds of receptors on the cell surface. They act as antennas to receive signals from the extracellular environment. These signals may be from endogenous sources within the human body, such as neurotransmitters, cytokinins, and hormones, or exogenous sources from outside environment, such as the viruses or drugs. On receiving the signals, receptors transduce these signals to the cell interior within the cell, and the signal may cause a cascade of reaction. I am discussing here a number of receptor classes and analyze how signals are transduced. These receptors are GPCR or G protein coupled receptors, ion channel receptors, tyrosine kinase, and intracellular or nuclear receptor. First, I am discussing about the G protein coupled receptor. Means the GPCRs represents positively the most important class of target proteins for drug discovery. They are always involved in signaling from outside the cell to inside. The number of diseases that are caused by a GPCR malfunction is enormous, and therefore it is not surprising that most commonly prescribed medicines act on GPCR. It is estimated that about 27% of drugs are designed to target the GPCR receptor. The common features of this superfamily of receptors, there are many different families and subtypes of receptors in this group, is that there are 7 domains that cross the cell membrane. These 7 transmembrane receptors are often referred to as serpentine receptors. The serpentine receptors are coupled to the G proteins, means the guanine nucleotide regulatory proteins inside the cell. There are 3 subunits that make up the G proteins: alpha, beta, and gamma. When a ligand, such as a drug or neurotransmitter, binds to a receptor on the cell surface, the shape of the receptor changes. This induces an activated change in the trimeric clusters of alpha, beta, and gamma subunits within the cell. Next one, that is the ion channel receptor. There are 2 main types of ion channel receptor, the ligand gated and voltage gated. In addition, some ion channels are regulated through GPCRs or via activation by amino acid. The ligand gated family consists of receptors of the so-called cis loop superfamily: nicotinic receptor, gamma aminobutyric acid receptor, glycine receptor, 5-hydroxytryptamine means 5-HT3 receptors, and some glutamate activated or anionic channels. The common feature is that they are made up of 5 subunits, that are the 2 alpha, 1 beta, 1 gamma, and 1 delta subunits. Natural ligands for this family of ion channels are acetylcholine, GABA, glycine, and aspartic acid. Voltage-gated ion channels depends on changes of transmembrane voltage to regulate the opening and closing of channel gates. A common feature of this type of receptor is the presence of 4 domains, where each domain consists of 6 membrane spanning regions. Some of these channels are the sodium, calcium, potassium channels, and they regulate the influx of these ions into the cell interior to propagate the signal. Diseases mediated through ion channel receptors include cardiovascular disease, hypertension, and CNS dysfunction. A voltage-gated ion channel is a key to the treatment of CF. Neurotransmitters binding to GPCR and ion channel. Acetylcholine binds to cholinergic receptor, of which there are two types, muscarinic and nicotinic. GABA binds to both GPCR and ion channel receptor. Dopamine binds to dopamine receptor D1 and D5, activates adenylyl cyclase, binds to receptors D2, D3, and D4, inhibits adenylyl cyclase. Norepinephrine binds to alpha and beta adrenergic receptors, causes vasoconstriction and increases blood pressure for the treatment of hypotension and shock. Serotonin binds to 5-HT receptor to act as excitatory and inhibitory neurotransmitter, as inhibitory function treats anxiety and depression, and excitatory function treats antipsychotic. Tyrosine kinase. This class of receptor transmits signals carried by hormones and growth factors. The structure consists of an extracellular domain for binding ligands and a cytoplasmic enzyme domain. The function of kinases is to enable phosphorylation. Phosphorylation regulates most aspects of cell life. When a ligand binds to the receptors and receptors dimerize and join together, this action activates the enzyme within the cell. As a result, protein molecules are phosphorylated. Other kinase receptors are serine, threonine kinase, protein kinase, and mitogen activated protein, means the MAP kinase, insulin transforming growth factor beta, TGF beta, and platelet derived growth factor, which called the PDGF, are the natural ligands that interact with kinase receptor. The last one, that is the intracellular receptor. Intracellular receptors are located inside cells in the cytoplasm or nucleus. These receptors can bind to DNA and regulate the expression of genes. Endogenous ligands such as hormones and drugs attach to these receptors to either activate, upregulate, or inhibit, downregulate transcription message from genes. Hence, these receptors are important in controlling the development of homeostasis, means the action of a system to regulate itself to remain stable and constant, and for metabolism. There is a large superfamily of these intracellular receptors. The family's common feature is to single polypeptide chain consisting of three distinct domains. Number 1, the amino terminus. Number 2, the DNA binding domains. Number 3, the carboxy terminus or ligand binding domain. The amino terminus, this region is most instances is involved in activating or stimulating transcription. Next one, the DNA binding domain. Amino acids in this region are responsible for the binding of the receptor to specific sequences of DNA. And number 3, the carboxy terminus or ligand binding domain. This is the region that binds ligand. So far, the 48 intracellular receptors have been identified and they belong to 11 subgroups. Some of the common receptors are estrogen receptors, glucocorticoid receptors, and progesterone receptors. So this is the summary of important points today's lecture 5. The number 1, the pharmaceutical companies evaluate the future direction and R&D activities on the basis of medical needs, market size, government policies and regulations, patent protection and key competencies. Number 2, most diseases apart from trauma and infections have origins in the genes or the proteins associated with them, as well as the biochemical pathways and networks. Number 3, current approach to drug discovery starts with the identification of target or targets that cause or lead to disease. Number 4, microarray is a technology used to study gene interactions and control of biochemical pathways. Number 5, target validation is necessary to confirm the validity of a target as a representative of a disease model before too much investment and time is expended on it. Number 6, once targets are validated, potential drug candidates are designed to bind and interact with these targets. Number 7. The main drug target are enzymes and receptors that are found on the cell surface or within the intracellular matrix. Number 8. Drugs interact with enzymes and receptors mainly through van der Waals forces and hydrogen bonding. They need the correct shapes and sizes to fit into the active sites of the target. Number 9. Drugs work in two ways as agonist and antagonist. Agonists activate the receptors whereas the antagonists deactivate, block, or inhibit the receptors. Number 10. After binding of drug and receptor, a cascade of signal transduction occurs within the cell and is manifested as a variety of effects on the diseased biochemical pathways. Number 11. The major receptors are GPCRs, intracellular nuclear receptor, ion channels, and tyrosine kinase. And at last, the assays are diverse to test biochemical systems in laboratories as they are readily available and provide a means to evaluate the effects of potential drug candidate.